Good afternoon, everyone. Thank you for taking the time to join us today for Cure Cancer Researcher Lunch and Learn with one of our current researchers, Dr. Susan Christo. A warm welcome to the Cure Cancer team, our members of our board, our committees, our observers, and obviously all of you, our donors, fundraisers, and supporters. It's great to see you here again. A little bit of housekeeping. This session will last around 45 minutes. And if you do have any questions, please type them in the comments section below, and I will weave them into the conversation as I go along. You may also notice a link to donate. If you are able to, please consider making a donation. Any amount to support our cause would be greatly appreciated. And we also have the opportunity to sign up for Change for Change, which is a third-party platform, and you can choose your charity of choice so that you make everyday purchases. The amounts will be rounded up to the nearest dollar, and that extra bit will be sent to Cure Cancer. Just simply scan the QR code in the background to be able to make a meaningful impact with your purchases. Finally, if you'd like to stay updated with our whole Lunch and Learn series, please don't hesitate to like us on Facebook. Our goal is to raise awareness about the critical need for cancer research funding, and your support means a lot to us. These small gestures can make a big difference in keeping the conversation going and driving progress towards a cure. Now, without further ado, let me introduce you to Dr. Susan Christo, who is joining us from Melbourne. Afternoon, Susan.
Good afternoon, Nikki. How are you?
Doing well. How are you going today?
Thank you very much. Yeah, very well. Thank you.
Good. Well, Susan is a senior research officer at the University of Melbourne and graduated with a PhD from the University of South Australia, one of my favourite places, in 2015. Today we will talk about her research in immunology with a focus on triple-negative breast cancer, the most aggressive breast cancer that sadly is more common in young women, and melanoma, which is often referred to as Australia's national cancer. Some big topics to cover today, Susan, but first of all, before we delve into your research, can you tell the audience a bit about yourself?
Absolutely. So you alluded to the fact that I haven't always been in Melbourne. So I was actually born here, even though I am currently living here. I was born here, and then we moved to your beautiful city, Sydney. So I used to live in Sydney for a few years before moving up to the Central Coast. And that was really sort of in my younger years before we moved to Adelaide, where I did most of my schooling. I did all of my schooling, rather, and my university studies. And it was around the time, I think I was 11 or 12. I remember being in year 7. And when people were asking, what do you want to do? Where do you want to go? And I always wanted to be a pharmacist. And that seems a bit odd now, but the reason I wanted to be a pharmacist is because I wanted to develop a drug to cure cancer. And I was so set on that. That's where I wanted to go. I've always had a passion for science. And so that really kind of set my path. That I was so determined to just carry forward with that. And so, of course, I took a lot of scientific subjects and mathematical subjects. And when it came to the decision, I actually chose to do a biotechnology degree, which is slightly different to my original thought, which was, pharmacy. But it really gave me the opportunity to go into research. And so through my biotechnology degree, I was exposed to immunology, the study of the immune system, which I think is the greatest topic in the world, bias a little. But through that, I was also really introduced to a particular immune cell, and that's called T cells. And although I'd love to say that T cell stands for terrific tumor fighting cell, it actually stands for thymus, which is a bit boring. But anyway, my passion for T cells really started quite early on in my career. And ever since then, we have really looked into the study of T cells across the body and across different diseases, which is where we kind of led into the cancer field.
Great. And you talked about immunology, and most people would know it's about your immune system. But can you, for those that aren't familiar, just explain a bit more about that term?
Absolutely. So as an immunologist, what we do is we study all things related to health and disease. So in our body, we have a collection of cells, which are broadly classified as our immune cells. And many people sometimes will classify these as white blood cells. So we often refer to those in the blood as white blood cells. But what's really interesting is that immune cells, they come in so many different flavors. I mean, every year I feel like there's just a new subset of cells or a new flavor of cell. And the particular type of T cell that we focus on is not in the blood, but they're in our organs. And we call these tissue T cells. Now, you might think, well, what's the difference between a tissue T cell and a blood T cell? Is it just because one is living in an organ and one's living in the blood? They're actually very, very different, not only by the way they look, but the way they behave. And we noted very early on, actually, when these tissue T cells were discovered, that they had actually a better response to infection and cancer. They were much faster and they were way more poised in that site to respond to these sorts of challenges. And so that really got us interested in understanding how not only our white blood cells, but really our cells in our skin, in our lung, in our gut, and all these different areas. Well, how do they make up our immune system and how do they contribute to our health? And really, I think the one thing that fascinated me about the immune system is we always think about it in the context of disease. We always think, oh, it's when it goes wrong. But our immune system is actually responsible for keeping us healthy. And a healthy person, that's not a default setting. Our immune system is working really, really hard to make sure that all of our organs are intact. There's no threat by bugs or cancer or autoimmunity. It's working overtime to make sure that we're really healthy. So just this collection of cells in our body are responsible for us being healthy and responsible for some diseases as well. So that just is an amazing and a lot of weight on their shoulders, but I think the immune system does a pretty good job at handling a lot of challenges that it sees.
That's a really nice way to frame it. So we don't give T cells or tissue T cells the credit they deserve. Is that what you're saying?
Absolutely. Yes. Thank you. I will advocate for tissue T cells because, every day, if you think about it, I mean, especially the T cells that are exposed to the environment, and I'm talking about the cells in our skin, the gut, and the lung, we're breathing in air, we're touching things, we're touching nasty bugs and dirt and everything. And they're working really hard to make sure that that doesn't lead to disease. So, we really care about tissue T cells because they're the first ones to see danger. The blood T cells, they take a little bit longer to get into the tissues. They take a little bit longer to kind of recognize danger. But this is why we care so much about understanding more about tissue T cells.
So a quick question on tissue T cells. Are they the same for every organ in your body or are they different?
Oh, that is such a good question because that's been a focus of our research for several years now. And we found that, in fact, no, tissue T cells are very different based on where they live. And it makes sense to us because at the end of the day, you really want your T cells to live in harmony with your organs. You don't want your T cells to be hyperreactive and cause autoimmunity, but you don't want them to be too slow and result in cancer. So there's a really fine balance. So for example, if you take a T cell from a skin, they're not going to be as fast as you would say in the liver. And the reason is because in the skin, they are dealing with so many pathogens, so many bugs and challenges on a day-to-day basis because, it's the environmental challenges that we see, they're touching things. So they need to make decisions. So they have a much higher threshold for when they decide this is danger. But in the liver, for example, anything that gets through to the liver is most likely danger. It's very unlikely that you're going to have a good bug in that system. So they're more likely to respond a lot quicker because they know that, no strangers allowed there. But the skin is a little bit more reserved. So already we see differences, but, it works. It's all balanced. It's all finely tuned, and it really works when things go right.
And just before we move on to your specific areas of study, how often do these cells regenerate? Is it quite frequently, or do they live for a long time?
So that's also dependent on where they live as well. So it's really a matter of whether the tissue is turning over very quickly. Things like what we call epithelial sites. So epithelial is essentially like layers of, brick layers, things like the skin and lung and the gut, things that have several layers. And so you might have cells that can regenerate quite quickly in that instance, but our tissue T cells, they can survive a lot longer in those particular sites. So they're actually very resilient. They can stay there for as long as they need to. So they're not, succumbing to all of this as fast turnover. And we think there's some different proteins in that environment that's helping them live a very long time, because the body knows that we need them. We need it because as these epithelial layers are shedding off, so to speak, you're losing skin cells every day. The ones underneath are seeing new pathogens. So you don't want to lose your immune system as you're doing that. So it's a very clever system, I think.
Yeah, sounds like it. Absolutely. So let's move on to one of the areas of focus, which is triple negative breast cancer. It is quite a feared cancer, as we know.
Yes.
We've got a very low survival rate, despite the significant progress in other types of breast cancer. I think the 5-year survival rate is around 97%, but not clearly for triple negative. So first of all, can you explain, why is it called triple negative?
So triple negative breast cancer, as you mentioned, is actually quite aggressive. It actually makes up about 15% of all the breast cancer. So it's actually a very big chunk of this kind of broad spread disease. And it's recognized as being negative or low for 3 particular proteins, the estrogen receptor, the progesterone receptor, and a growth factor. And so this particular tumor cancer is quite aggressive, mostly can occur in younger patients, which I think is one of the kind of concerns of these sorts of cancers, is because it comes up a lot earlier than other types of breast cancer. And we're talking about premenopausal ages, sometimes under 40 years old. So, it's quite damaging for a lot of young women who may succumb to this particular disease. But and unfortunately, there's also a high risk of metastasis. So, the idea that this tumor can spread around, and because of that, it's definitely considered an aggressive tumor.
And a question here from the audience, and thank you. If you do have any questions, please drop them into the Facebook chat. So why has there been less progress in this type of tumor research than that in other breast cancer tumors?
Oh, that's a great question. I wish I had one particular answer, but I think it really comes down to a multitude of factors. And I think that, one of our biggest questions that we have in the lab is, why doesn't a patient respond to common therapy? And that's something that we're always trying to address. And it's really hard to actually pinpoint why. It could be environmental, it could be genetics, and it may be a multitude of factors. So I can't really give you one particular answer, unfortunately, but we're certainly working on understanding what's common between all of the patients, so we can target that process instead and try and get a more equivalent responsiveness across the patients. In the case of triple negative breast cancer, something that we've come across is that there's a lot of these tissue T cells in that environment. And we didn't really know that for several years, because nobody really looks at that, and nobody has really understood what a tissue T cell is for a very long time. It's a relatively new population, actually. And so we're starting to now ask these questions about whether the tissue T cells are actually the answer to whether these ones can be targeted, so you do get responsiveness across different tumor subtypes and across patients, and you get this really nice homogeneous response.
Okay. So in traditional therapies and treatments, it would be radiation or chemotherapy. In some cases, men and women, because men can also get breast cancer, have hysterectomies. So what's your focus in regards to triple negative and the impact that those tissue T cells can have?
Yes. So definitely radiation therapy is one of the most common treatment types, but not only in breast cancer, but in a range of different cancers. And it's almost been a gold standard for decades now, because it's really effective at shrinking a tumor. And when a patient presents with a tumor, the main objective is to get it as small as possible, as quickly as possible. So patients might undergo what's called a resection, which is the removal if it's possible. And in some cases, they may turn to radiation therapy. And what that is just really high energy of irradiation that essentially damages the DNA of the cells. And therefore they die, they break up, and you kind of lose that tumor structure and you begin to see that shrinkage. The problem with radiation therapy, although it's very effective at shrinking a tumor, is the fact that it's what we call non-specific. It's not specific to a tumor, and it also will affect healthy cells around that site, including our tissue T, sorry, not including our tissue T cells, but including our immune cells. But one of our very exciting findings, I jumped the gun because I was just so excited, is the fact that amongst all the immune cells that are exposed to this irradiation, it's our tissue T cells that don't die. And so I like to think of it as a really heavy rainfall, rain and thunder, that's the irradiation. And somehow these tissue T cells are carrying this super umbrella, and they're not getting wet and they're not dying, and they're seeming to survive this irradiation process. But not only that, after the irradiation, they're still able to perform their functions, which is to protect and to shrink a tumor. So now we're starting to realize that actually we haven't necessarily been looking at the full picture. We've kind of just taken a really full-on approach of just getting that tumor as small as possible. But now we're starting to recognize that there might be some little gems in there, and we want to understand what's this umbrella, what's this molecular umbrella that these cells are holding that are protecting them from the irradiation, and how can we give some other immune cells this umbrella so we can protect our good cells whilst shrinking our bad cells. And yeah, that's really one of the focuses of this project that we're looking at, and we really hope that it's applicable across a range of different tumors as well.
No, that would be amazing. I'm just going to pause there and welcome some of our new guests. Thank you. We're talking to Dr. Susan Christo, who focuses on immunology from the University of Melbourne. And we've just been talking about irradiation therapy and triple negative breast cancer. If you do have a question, please drop it into the Facebook chat and I will incorporate it in our discussion. So you've talked about this umbrella, if you like, and the T cells, the tissue T cells being protected. So you've start to see this happen. Where do you go from here in your research? What's the next phase of your research?
We're trying to understand exactly how they are protected, because when you're exposed to radiation therapy, a lot of things happen. You change your genes, you change your proteins, but that doesn't seem to be happening in the tissue T cells. So what we're trying to understand is what's their protective mechanism, so to speak. What's the protein? What's the gene? The so-called umbrella, what is that that's shielding that cell from any sort of damage? And what we hope to do is move forward with designing some sort of therapy, perhaps a drug or a sort of target that can be used to, that can be administered to patients in conjunction with radiation therapy. So I think the important point here is that we want to make it better. We're not trying to reinvent the wheel. We want to make it better because we know it's effective. It's a really good way of shrinking tumors, but how do we protect our good cells in amongst the mix of that? So developing a drug target that can protect immune cells while allowing the tumor cells to die, I think would be our hope for the ongoing future.
Yeah, it's almost like a double threat then, isn't it? You're killing the cells.
Yep.
You've got these very powerful, strong cells that continue to grow.
Absolutely.
You talked about how this happens in triple negative breast cancer. Has that happened across other cancers, tumor types as well that you've seen this in?
Yes, absolutely. So obviously radiation therapy is across different tumors, but we also know that tissue T cells are present in other tumors as well, including melanoma. So, I mean, I spoke a lot about the skin. We're actually skin people. We love the skin. So we tend to look there a lot, but they're definitely surrounding that melanoma. They're within the melanoma, and it's something that we think could be a common factor that we can be targeting across a range of different types. But also more and more data keeps coming out every month, it seems, in the literature, that the more tissue T cells you have, the better the survival of the patient. So we're starting to see numbers now. You know, this is not just an observation in the laboratory. We're talking about large scale patient cohorts where we can take thousands of patients and we can look at the types of cells in their tumor and we can start to stratify them. And we really see that patients that have more tissue T cells, do a lot better in terms of their 5-year survival and their prognosis. So I think there's a really exciting avenue for us to explore here that we haven't really fully understood in the past.
So this would take me on to a next step where if you've got more tissue T cells, that's a good thing.
Absolutely.
Is that just genetics, or how can you acquire more or encourage more?
Yes. That is a good question. And so this is something where, again, there's so much research into how do we push that pathway? How do we sort of, whisper in the ear of the T cell and tell it to become a tissue T cell? And we're doing a lot of work in the lab now where we can manipulate cells. There's a whole field of cancer treatment called immunotherapy, which I'm sure a lot of the listeners may be aware of. And that's the idea of targeting the immune system in some sort of therapy. So it could be something like what's called checkpoint blockade. It could be something like adoptive cell transfer or CAR T cells. Again, these are quite trendy words at the moment, but it's this idea of stimulating the immune system. And what we want to do is we want to take our existing T cells in the blood, for example, and we want to give them like a roadmap and tell them this is where you need to go get into the tissue. Because at the moment, one of the biggest factors actually is that the T cells can't quite get into the tumor. So getting into the site is also another challenge for a lot of these different types of immunotherapies. But we're seeing a big surge of cancer vaccination. So the idea that you can vaccinate not only against bugs like we do for COVID and flu, for example, but now beginning to see whether we can vaccinate against a cancer. And that is such an exciting area, which is, it's just really opening up lots of possibilities. But whether we can incorporate what we know about, tissue T cell biology into these formulations and say, well, if you add this protein or you add this gene, you can make your cell become a tissue T cell through this vaccination process. So I think there's a few avenues that we can try, but it's definitely an active research area in the laboratory.
Yeah, absolutely. And there was an announcement a couple of months ago about the new vaccination for melanoma for certain patients with it. So, I mean, you talk about progress. I mean, it's phenomenal to think in 2020.
Absolutely.
We've got these options now that you would never think were possible. So just shows you the power of research. So thank you. You've talked about large cohorts. You've talked about the power of the tissue T cell. I think we're all fans here now. With your research, obviously there's a lot of research going on. Lots of researchers are focusing on immunology. Do you do any collaboration? We obviously have 503 researchers that we're funded.
Yeah. Yeah, absolutely.
On this big problem you're trying to
yes.
solve.
So this is, I think, one of the joys of our job, actually, is the collaborative nature. So science, I will always say, is a team sport, and it's an evolving game. And I think that we will never progress without coming together. And in fact, the best example of that was COVID. And we saw scientists around the world come together, and we had a vaccine within a year. I mean, that is phenomenal speed in the medical research world. So, you can only imagine what we can do when we come together in the cancer field as well. And so we collaborate with some amazing scientists at the Peter MacCallum Cancer Centre, particularly Paul Vivas, which I know is a big cure cancer fan of, and big fan of cure cancer, and as well as Shireen Loy, who is a clinician over at the Peter MacCallum Cancer Centre as well. And I think having this mix of scientists and clinicians coming together, solving these issues, and really from our perspective, we do discovery research. We're what we call fundamental biologists or immunologists, and we're here to find immunological solutions for clinical problems. So it's about having these discussions about, well, what's really going on in the clinic. I mean, you can read papers and you can try and get an understanding, but it's actually speaking to the people and getting a sense of the reality of the situation and the humanity of the situation as well. So working with Paul and Shireen, for example, has just been so enlightening and amazing, because they bring this experience of translation that from bench to bedside. It's such an amazing team. So we'll always advocate for collaborations. And actually, Nikki, if I can jump in here and also state that, we talk a lot about scientific collaborations, but I also want to highlight that we work as well with our consumers. I really want to credit our consumers at the moment, and I won't name them for privacy, but we have 4 amazing ladies who have generously given up their time to speak about their lived experience of cancer. So most of them have had breast cancer. They've also had a radiation therapy. And, you know, they speak about this rather traumatic time of their life, and they give us a real sense of just the urgency, and, you know, it just motivates us more and more. So I really want to highlight that. And I really appreciate our consumers. I want to say thank you to all of them if they're out listening. And yeah, it adds such wealth of knowledge to our team. I think it's critical to have that lived experience in our team.
Yeah, look, absolutely. And they're also ambassadors for progress in research as well, because
Absolutely. Yeah.
they've lived through it, they've been through it, and they can give a unique first-hand experience of it too. So
yes,
really important. And likewise, we talk very few of our researchers are clinicians, but we do have a couple, and it's, you know, the important role that clinicians play with research as well, because they're dealing face to face with patients every day too.
Absolutely. Yes.
I would say it's definitely a team sport. So
it 100% is, yeah.
Let's get into the nitty gritty then. So what do you do on a day-to-day basis? Are there something you do every day? Are you in the lab all the time? Or are you not? Talk about your traditional day.
Yeah, absolutely. I think every day is very different depending on what's going on. But if you were to categorize it, our work involves three main things: experiments in the lab, a lot of reading, and a lot of writing. And so, you know, we balance a lot of these things in different proportions every day. But essentially what would happen is, for example, I would grab a sample in the morning. I'd have two groups. I'd have a group that has been irradiated and a group that hasn't been irradiated. And I'd go down into the lab and I'd process these tissues. And then at the end of the day, I'd run it on a machine, a very fancy machine that can tell me how many tissue T cells I have left at the end of the day. And I can look at different proteins and genes and do some really high-end and multi-dimensional analysis on these particular tissues. And so, you know, that we can do different variations and test different things and add different drugs and see what happens in these instances. And along with that, we're doing a lot of reading and just getting ourselves as much up to speed as possible with the literature and what's happening, and trying to understand where the field's at, where the field's going, how does that fit in with our work. And of course, we really enjoy presenting our results and writing about it, because we want the world to know about how awesome tissue T cells are. And so that really does factor in as well to our time. So it's really a mixed bag of what we're doing on every single day, but we definitely love getting those results and then communicating those results. So we're always looking for opportunities to chat with anyone. If anyone's interested, please reach out to us. We'd love to talk about what we do. We'd love to take you through the Doherty if you're in Melbourne. And so it's just fun. It's just the most amazing environment. I love my job.
Well, you sound like it. You know, I've way research is a team sport. I've learned how awesome tissue T cells are, and you sound like you're having, you know, dealing with serious issues, but making great progress, which is wonderful. Just going to jump in here. We've got about 10 more minutes with our chat with Dr. Susan Christo. So please, if you have any questions, drop them in and I'll feed those in. So you are one of our alumni for this year. We are funding you as an early career cancer researcher. Could you talk a bit more about what that funding means to you?
Oh, it meant the world to me. And every time I speak with your team, I'm just in awe at the work they do, because it's just one of the most rewarding experiences of my life. I find that from the very start, actually applying. So as researchers, we need to apply for a lot of funding, mostly from the government. But in cases like these sorts of organizations, they do an amazing job of helping young researchers like myself really get off the ground and get this idea blooming, certainly in the cancer field. But, you know, what I loved about the Cure Cancer process was the focus wasn't just on, you know, give me exactly when you're going to find protein A or protein B. It was about the significance and the translation of the project. And I actually really enjoyed the application process, because it gave me a chance to sit back and say, you know, this is exactly where I want to go, and this is where I want to be, and this is what I want to achieve. And so I think having the scientific focus on the outcome, as opposed to just tick boxing, for example, which is most applications, they're very long, they're sometimes up to 40 pages, whereas just to have like a really, really direct discussion and overview of what you want to achieve and how you're going to do that, I think was just a really nice process. So I commend you and your team, Nikki, because it was a great experience, and it really means a lot that, you know, I've had this support throughout my career to build up this work and hopefully see it being translated into the clinic.
Look, no, absolutely. I mean, for us, we brought our research grant process in-house a couple of years ago, and to make it easier, simpler, and more transparent for researchers to apply for grants is what's important. We know from many of our researchers that you spend a long time trying to apply for grants, and your success is often very low. So
yeah,
an easier way that we can help fund brilliant research like you are doing, then that's what's important to us as well. Now, just looking.
So yeah, thank you very much to all the donors and fundraisers as well. I mean, it means a lot to us. We really do appreciate your support.
Yeah, look,
for these sorts
absolutely,
of
and I
projects.
know they appreciate your work. I've just seen a quick question by Sarah. Sarah, if you want more information, please send us an email at info@curecancer.com.au, and we'll get back to you with some further details. Okay, thank you. So just as we start to wrap up, one question about tissue samples. Now, are they all from Australia, or are you dealing with samples from people overseas as well?
We mostly deal with Australian samples, and the reason that we do need to use more local samples is because for the work that we do, we often like to work with fresh samples. So, I mean, a lot of the times we can, you know, surgeons can take a piece of a tumor or a piece of a tissue and throw it in the freezer or throw it in liquid nitrogen, and then we can come back to it several days or weeks or months later. But in our case, we really like to work with the fresh tissue. So a lot of the time we have to work quite closely with clinicians and surgeons and other doctors, oncologists that are dealing with these patients, and we're coordinating a lot of these processes to try and get that tissue so we can work with it as fresh as possible. But yeah, so we definitely, like I mentioned, have collaborations with the Peter MacCallum Centre. We also work with the Austin Health in Melbourne as well. So we try and really just broaden our kind of sampling base so we can get all the samples and really test a lot of our research in the human side of things as well.
Yeah, look, that makes sense. Absolutely. So from your perspective, where to now? You've identified these cells that have an umbrella to protect themselves. You're looking at a more effective treatment along with radiation therapy.
Yep.
So what's the next step for you in your research?
So of course, I'd love to identify what the umbrella is, what that gene or protein is, and then also start to ask whether we can drug that target, or, you know, make a drug to replicate what we see in these tissue T cells in other cells as well, and really improve that immune response. But another kind of arm that we're doing in the lab is also looking at immunotherapy. So coming back to this idea of targeting the immune system and understanding, you know, again, why do some patients respond to this and why do others not? And can tissue T cells be that common factor that we can target? Is there something on that tissue T cell that makes it stand out? Can we use an immunotherapy that can target that cell to help it make it more superior, to make it more like supercharge it, if you like, and make it more robust killer in that tumor environment? So we're asking all these different questions in a few different ways, and we're really hoping that a lot of our work will be translated into the clinic in the future.
Look, it sounds like you're making great progress, and thank you for, on behalf of everyone that's listening as well, for what you are doing. It's really important, particularly in the fight for triple negative breast cancer, and trying to find a better treatment for those people going through that.
Absolutely. Yep.
So
absolutely.
Before we wrap up, any last words, anything you want to talk about that we haven't yet covered in chat today?
Oh, just a general thank you once again to Cure Cancer, to the donors, to the fundraisers, for everyone who support, because at the end of the day, we like to think about, you know, medical research as a pipeline, and that pipeline starts with discovery research. You don't get the clinical outcomes unless you have fundamental understanding of biology. And there's a real concern at the moment with Australia not really focusing on that step one, if you like, of this pipeline. And the more that we cut medical research we're not going to start to see the discoveries. And that's really going to affect us, maybe not next year or the year after, but it's going to start affecting us in 10, 20 years. And as our population is growing, as cancer is becoming a lot more prevalent in this population, we need to step it up. And we thank you again for helping us with that discovery research, because that is really what's driving this pipeline. And so again, thank you very much, Nikki, for the opportunity to do our work.
Not at all, and you say it very wisely. I mean, that's what we always repeat, that without a pipeline of early career cancer researchers, you're not going to get the more tenured professors. So
Yep.
It's vital we continue to fund the brilliant work such as yourself and the team there are doing, to find new ways, innovative ways of trying
Absolutely.
to
Yep.
combat something that we haven't yet found a solution for.
Yep.
So,
absolutely. We're willing and ready to outsmart this cancer and eradicate that from the population, make it a manageable disease, because it's a real burden on so many Australians and so many people around the world, and really an emotional process. So we want to provide hope for any Australian who's dealing with this and suffering through. So we remember you, we're motivated by you, and we wish you well on your journey.
Oh, lovely. Well, thank you, Susan. It's been great to talk to you. Thank you to all our audience as well. I hope you found that informative and insightful. I certainly want to learn more about tissue T cells now. They sound like
anytime. Come down to Melbourne, we'll show you.
I will be there. So thank you. For our audience out there, we're next speaking to Dr. Eric Kusnadi on the 14th of September, also a Thursday at 12:30. He's at Peter MacCallum Department of Oncology, also in Melbourne, and his focus is on prostate cancer, and we'll be marking Prostate Cancer Awareness Month, where he'll be sharing the latest in his research and talking about the hopes for the future treatment of prostate cancer as well. So thank you for joining us all. This will be available on YouTube, and we'll also email it out to you next week, so you can all have this recording. And any questions, please do drop us a line at info@curecancer.com.au. Thank you. Have a good afternoon.
Thank you.